Thank you everyone for listening to my talk. I'm Sabrina Heiser, and I'll be talking about strong genetic differentiation and selfing shaping the population structure of a red macroalga ecosystem engineer. The genus of our study organism, Plocamium, has been particularly well studied at Palmer Station on the western Antarctic Peninsula. The main focus of past research has been on its chemical defenses. Plocamium is often abundant and known to support one of the highest mesograzer abundances compared to other seaweeds. Yet with one exception, none of the grazers will actually feed on their host. Young et al. Published a study in 2013 looking into the chemical defense diversity of Plocamium around Palmer Station, and they found 5 different chemogroups. A chemogroup is basically a signature mixture or a signature cocktail of different secondary metabolites. And as you can see from the examples here, chemogroups differ in the compounds they have present and also in the relative quantities that they are producing them in. And since the original study, we identified around 15 different chemogroups, which we arbitrarily named A through O. We used the COX-1 gene for barcoding in order to determine whether we were dealing with more than one species. As you can see from the haplotype network on the right, we found 2 main haplotypes, 1 and 2, which also had previously been described by Young et al. As indicated by the different colors, we can also see that the chemogroups sorted themselves quite well into the 2 different haplotypes. So how are these chemogroups distributed around the different sites? To describe the fine-scale distribution of the chemogroups, we run transects perpendicular to the shore at 8 different sites around Palmer Station. And these are the results for one of the sites that we surveyed. On the horizontal axis, we have the number of thalli, which are color-coded by the different chemogroups that we described or found. And then on the vertical axis, we have our depth distribution that we collected from. And when we compare across the sites that we surveyed, we see that chemogroup composition differed significantly between the sites and sometimes even between the depths. And the question is whether genetic differentiation can explain the distribution of the chemogroups that we are observing here. To answer this question, we developed microsatellite markers, which are repetitive motifs that generate inter-individual variation. We analyzed the samples for genetic structure by site, and we weren't really able to find any clear patterns. And considering that we previously found that chemogroups were haplotype specific, we decided to look at genetic structure by chemogroup. And as you can see, based on the discriminant analysis of principal components, we see that principal component 1 explains over 99.9% of the genetic variation between the samples, with chemogroup L forming one cluster and the other chemogroups forming a second cluster. And this is very intriguing, considering, looking back at the distribution of the chemogroups, that we found some of these were collected within close vicinity of each other. When we described the reproductive system of Plocamium around our sample sites, we found a predominance of positive FIS values, which indicates an excess of homozygotes and therefore high levels of selfing. To summarize, we found a strong genetic differentiation between chemogroup L and the other chemogroups, which also shows that morphology and even barcoding alone is not necessarily enough to assess species diversity. We also found high levels of selfing, which we know can lead to high homogeneity, genetic homogeneity within sites, and at the same time high differentiation among sites. And this requires further investigation, since it could explain the distribution patterns of the chemogroups that we can observe. We also know that sites differ in the amount of disturbance and currents that they're exposed to, which is another factor that may contribute to within-site distribution patterns of the chemogroups. Overall, this shows that to understand the distribution of intraspecific diversity in an important ecosystem engineer, we need to take its genetic structure, reproductive system, natural history, as well as phenotypic traits into account. With that, I'd like to acknowledge everyone who has been absolutely instrumental to this research, especially our funding sources such as NSF, the Antarctic Support Contract, and our field teams. And I'm happy to take any questions.